Okay, do you see that thing right there? That means it's recording. Okay, that means that you don't have to interrupt me and ask me if it's a recording. Okay, good. So it's doing that, but I do forget because it's not particularly important to me and it's important to you. Like, you know, my job's done when I'm done here. Okay, so where we were last time is we were talking about basically a lot of physiology being about exchange, getting things from one place to the other, knowing how it's exchanged, why it's exchanged, what would happen if you didn't. Okay, and now we're going to talk about the medium through which it's exchanged, which is primarily fluid, right? We are around 60-ish% water, a little more sometimes, a little less sometimes, okay? So, but the water is sequestered in multiple ways. So let's talk about that concept. So we have in about a 150-pound person, it's pretty damn impressive. You got about 42 liters of water. That's like, you're mostly water. That 10 pounds that you needed to lose, it's just water, guys. It's just water. So you just feel good about yourself. But the water is sequestered, and it's not all identical to all other water. So if you take that 42 liters, that's considered total body water. That's an abbreviation that isn't very frequently used, but your textbook uses it. But importantly, the total body water is sequestered. So there are body fluid compartments. And the body fluid compartments are extracellular fluid and intracellular fluid. And then there are some subdivisions. First off, when you took 1610, instead of referring to it as intracellular fluid, you usually referred to it as what? Cytoplasm, cytosol. I don't know why, but in phys, it's always intracellular fluid. So we're talking about the same thing, right? We're talking about the fluid that's actually inside the cell. So it's ICF, intracellular fluid. Now, note to self, something that you know. What does the prefix intra mean? Within or inside, okay? And extra, of course, means outside. And then we will also run into inter in a minute, and that will matter too. Okay, so ICF is intracellular fluid. Please note that that makes up the vast majority of the fluid, right? 28 of the 42 liters is intracellular fluid, good? Okay, but then you have extracellular fluid, and the extracellular fluid is the fluid that is outside of cells between them, right? And the extracellular fluid has a further subdivision, which is important. So this is another compartmentalization. The extracellular fluid is divided into plasma. And plasma would be extracellular fluid that surrounds what kind of cells? Blood cells, exactly. And it's often called just blood plasma or plasma. And it's circulating, so that'll be important. Okay? And then the rest of your extracellular fluid is mostly, they call it ISF, but that's also not a very common abbreviation. Interstitial fluid, often called tissue fluid. So I want you to picture from anatomy where this would be. Picture areolar connective tissue, right? Areolar connective tissue. And areolar connective tissue has cell, a bunch of fibers, and then other stuff that you typically refer to as matrix. Now what I'm telling you is that matrix is the vast majority of it is interstitial fluid, tissue fluid. That's like around cells that are not blood cells, okay? All right, so importantly, what does the prefix inter mean? Between, exactly. So inter, if you wanted to get out of Utah, what kind of highway do you get on? An interstate highway, right? Get the hell out of Utah. Okay, so interstitial fluid. Okay, now, these are compartments, but they have to have exchange. We know that you can't have them be solid compartments without the opportunity for exchange. Otherwise, right, the glucose that you took in when you ate your Snickers bar could never get into the little, the cell of your little finger, right? So we have to have a barrier and opportunity for exchange. So you guys already know this. What is the physical structure that is the barrier that controls exchange between the ECF and the ICF? What is the physical barrier that controls exchange between the ICF and the ECF? Cell membrane? Yeah, it's the cell membrane, right? And pretty damn important because, of course, you don't want your nucleus falling out, right? You need more glucose inside than you need outside. You got to make sure that you get rid of your carbon dioxide. So the ICF and the ECF are different, right? And the cell membrane controls transport between them. So you do have the opportunity for controlled transport between them, right? And you spent 1610 learning about that. Now we're going to apply it to this, really. Okay, and then likewise, there is exchange between the blood plasma and the interstitial fluid. And this one may be a little harder to come up with. But there is a physical barrier between the blood plasma and the interstitial fluid. Does anybody know what it is? What controls, what physical structure controls or allows for exchange between the blood plasma and the interstitial fluid? Look at this picture. I'm in the interstitial fluid now. I'm in the blood plasma now. Yeah, it's the capillary endothelium, yes? Also called epithelium. Most people are comfortable with epithelium. Endothelium is just a special type of epithelium. Okay, so, and remember that this has varying levels of permeability depending on the capillary structure, right? Remember, some capillaries, like the ones in your blood-brain barrier, are damn near impermeable, right? But then you've got fenestrated capillaries, which are more permeable, and then you've got sinusoid capillaries, which like almost anything can get in and out. So that's variable. So because there is possibility for exchange, you can do what we talked about before, right? You can get something from the interstitial fluid to the blood plasma, back out of the blood plasma, and then ultimately into the cell, right? Okay, so these are body fluid compartments. Now, the ICF and the ECF, for instance, have to be different, but they have to maintain exchange between them. And it's difficult to allow for exchange between things and not have them become the same, right? So for instance, in your home, if you want your home to be 72 degrees and it's 100 outside, right? And you've got children, you're going to leave the damn door open no matter how many times you say it. You know they are, and you did it too, okay? Then what's going to happen is the exchange makes it a little bit problematic maintaining homeostasis. So for instance, you're going to have to use a bunch of energy to maintain the difference between the external and the internal environment. That makes sense? So a lot of homeostasis is going to be making sure that you allow for exchange, but also allow for the ICF and the ECF to be unique. For instance, the blood plasma should have clotting proteins in it, and the interstitial fluid should not. The ECF should have really high concentration of sodium, but the ICF should have a really low concentration of sodium. But what does sodium want to do if it's high to low? Yeah, it wants to move, right? So you're going to have to work on this, right? To maintain both transport capacity and also difference between the things. So we'll start talking about that today. Okay, so what kinds of things do we work on transporting and maintaining composition of? The ECF and the ICF basically have different concentrations of nutrients, gases, wastes, and ions or electrolytes. And we work really, really hard at maintaining the ideal concentration inside and outside the cells. And it's relatively energetically expensive. Now, these, of course, you're familiar with what most of those do. You know why you need oxygen inside the cell. You know that carbon dioxide is produced as a byproduct of metabolism. You know why you need glucose inside a cell and amino acids and those kinds of things. You probably understand what some of the waste products would do if they built up. But the thing that usually people are least experienced with by the time they get to phys is the importance of ions, which if somebody's trying to sell you something, they'll call them electrolytes, right? So why do you work so hard at keeping potassium concentrations high inside the cell and low outside the cell? Sodium concentrations high outside, low inside. So that's what this class will really, really solidify if you didn't get it in other classes. What the hell do ions have to do with anything, and why do you worry about those kinds of things? Okay, primarily sodium, potassium, calcium. Okay, how important is this? How important is maintaining both transport and also this sort of unique identity and characteristic of each one in a cell? Each individual cell spends about 60% of its energy maintaining the homeostasis of these concentrations. It's incredibly energetically expensive to be really different from your external environment, right? So if sodium is really high out there, and you don't want it to be high in, what do you have to do every time it leaks in? You have to pump that crap back out, and that costs you something, right? Just like in your home. Really expensive to be 72 inside and 100 outside. So you spend about 60% of your energy regulating the composition, volume, temperature, pH of your fluids in order to maintain homeostasis. So I have a concept question for you, and it doesn't matter if you get it wrong because you're going to understand it by the time we talk about it. I'm going to tell you that the body concentrates primarily on one of these two, assessing and regulating and fixing the composition, temperature, pressure, pH of either the ECF or the ICF. Anybody want to hazard a guess as to what the body is really, really tightly controlling? Go ahead. ECF. Anybody want to hazard another one? It is the ECF. Now, there's conceptual reasons that you might guess ICF, right? Because it's pretty important to maintain the homeostasis of the cell. But I want to tell you that you have, on average, an adult person has 100 trillion cells. That's 100 trillion independent ICF pools. Now try to maintain every damn one of them independently. It is not economically feasible to maintain every one of them independently. And in addition, if you waited until a change got to the ICF, it might be too late to protect yourself from a pathophysiological condition. So if, for instance, I ate a poison, look at this situation. Oops, ate a poison. Where's it going to go first? ECF or ICF? ECF. Do you see that that's probably a better place to actually detect and try to fix any kind of change or disturbance, right? So it's both more economically feasible, right? A better use of your energy. It's also more protective because if changes are massive in the ECF, they will eventually affect the ICF, and then you have a pathophysiology situation. Does that make sense to you guys? So what we have, now I'm not saying you don't, cells do care about their own homeostasis, but collectively, if I'm monitoring body temperature, am I monitoring body temperature mostly in every single cell, or do I have, for instance, temperature receptors inside my bloodstream detecting the temperature of my plasma and detecting the temperature of my tissue fluids, right? And then what I could do, hopefully, is elicit this sweat response before, for instance, I actually change the temperature of my brain cells. Does that make sense? Okay, so what you are primarily going to work on is the ECF to protect the ICF, right? Okay, so if the composition of the ECF changes massively and you don't catch it, then you'll affect ICF function, right? And then you'll affect how actually the cells are functioning. Okay, now, do I or does anybody know everything about human or animal physiology? Absolutely not. What we do have is a phenomenally strong predictive framework that we're constantly adding back to. So science is not a body of knowledge, science is a discipline. Science is a way of figuring out when you were incorrect and a way of adding evidence when you're correct. So are we going to figure out something from now until your career is over about physiology that we didn't know? Yes, in all likelihood. Is it very, very likely to overturn the body of knowledge about how the cardiovascular system works? No, it's not likely. So what you're going to do is continue to hone and add to, which is why I hope my medical professionals are obsessive information researchers and take in information and continue adding to their body of knowledge. And I also reserve the right to tell you if something is wrong in your textbook, right? I will try to fix it if there's better knowledge out there, if there's new research. Okay, so now every once in a while you'll see this. I don't know why it's upside down. It's supposed to say IP, interactive physiology. So interactive physiology is, as I mentioned to you guys last time, it's part of the online content that comes with your textbook. If you know you're a really visual person and really need to work with things in order to see what happened, and I need to turn something up and see what happens, then that might be worth you investing in. And also, like I said, I think I can find access to a free online version of it, but I haven't had a chance to look yet. But in your notes, sometimes you'll see that. That means that I know that that particular region of interactive physiology is really helpful. Do I test out of it? No. Is it a really good way to know whether you're actually getting something? Yeah, it is. So it'll be there for you if you want it. And if I find the link, I'll show you the link to the free version. Okay, so ideally what we are going to try to do is we're going to use as our best prediction for what the human body will do, homeostasis, right? We're going to try to maintain a relatively constant internal environment despite what the external environment is doing, right? So next thing we're going to get to is how do you do that? How do you maintain a relatively constant internal environment? The primary mechanism that you use to maintain homeostasis is something called negative feedback. And in order to understand negative feedback, and you probably heard it before, you need to dump your former association with the word negative, whatever connotation of bad you have, right? And also, it doesn't mean lower than or less in this context. What it means is opposition. So, for instance, in a negative feedback situation, in order to maintain homeostasis, if my blood pressure goes up, then the prediction, of course, to maintain homeostasis is that negative feedback would bring it back down. But if my blood pressure went down, what would negative feedback do? Bring it back up, right? So do you see that it's not directionality? Negative means negate or oppose, right? So what you're going to do is whatever direction the disturbance is, negative feedback opposes it to bring it back into homeostasis, okay? All right, so it means that any deviation from the normal value is resisted or negated. And it's returned where? So this conceptually, maybe you know, maybe you don't. So let's talk about body temperature for a second. You have in your head 98.6 degrees Fahrenheit for a normal human body temperature. But my question to you is, if my body temperature gets disturbed, like if I go running in the heat of the day, my body temperature is obviously going to go well above 98.6. And I'm going to try to bring it back down, obviously, via negative feedback. Am I actually heading for 98.6, that set point, or is there an acceptable range? Yeah, there's an acceptable range. It's like in your home, right? If you have your thermostat set to 72, when it goes up to 72.2, does the air conditioner come on? No, because nobody can afford that, right? Okay, so you got it set to 72, but there's actually an acceptable error range about 72, right? So what you're doing in the human body is actually you have an acceptable error range for about every variable, right? You've got 98.6 maybe set, but maybe not. And then it's okay at 98.7. It's definitely not okay at 103, right? Right? It's not okay at 92. So you're actually dealing more with ranges than set points. So let's look at a graph of negative feedback for a typical human. And let's talk a little bit about graphs, because you guys are going to be doing graphs. Either you did them last week or you're working on them this week in Phys. Phys has less cool pictures than anatomy. There's going to be a lot of graphs and there's a lot of flow charts, because it's about functionality, right? More functionality than actual structures. So first thing is you have to know that graphs say specific things based on how you set them up. So let's just make sure that you understand that the x-axis and the y-axis are not interchangeable, because if I set up a graph, the x-axis is the independent axis, and the y-axis is the dependent axis. So let's say that we are going to do body temperature over time. Now, why does this matter? Because if I set up the graph this way, this says that my body temperature varies over time. If I set it up the opposite way, you are saying that time varies as my body temperature varies. And you don't have that superpower, do you? No, and it would be a sucky one. You're like, this is what I got. I'm a mutant, and I got this. Okay, so that makes sense? So I want to talk about what negative feedback looks like over time. So a negative feedback graph, what it tends to do is, let's say you knew, you don't generally, but let's say you knew that your ideal body temperature was 98.6. You know that we're not all 98.6. If you live with another human, you know that our body temperatures vary dramatically, and you can have thermostat wars with your significant other, right? I have no idea how that got turned down, honey. I think the dog did it. So let's say mine, which is, I like it cooler than my husband. My body temperature is maybe not 98.6, maybe it's 98.8. So I want it a little cooler in the house to cool me down. I don't know what it is. His is different though. But I'm not going to stay at that body temperature, right? It doesn't really happen. So what's going to happen over time is over the course of the day, you are going to go above and below that body temperature. If you take enough data points, what you will get is a really good idea about what my ideal body temperature range is. Does that make sense? So over the course of a typical day, let's say this is somebody else, and you're going to have a day in which you accomplish every damn thing you meant to. Remember that one day that happened, right? Remember? Okay, so here we are. What do you predict about your body temperature before you wake up? Lower. So activity and metabolism are the primary ways of increasing your body temperature, right? So metabolism can go up, or activity, or both can go up. So as you wake up, of course, your activity and your metabolism are going to go up, and your body temperature is going to go up. So maybe you were below ideal, this body temperature. Then you're going to get up and you're going to have breakfast, and then your metabolism is going to go up, right? Okay. And then you're going to sit down for a minute, plan out your day, and then this is one of those ideal days. You are going to work out before work. Oh my gosh. I don't even know how you humans do that. Like literally it feels like my whole house is on fire before work. I don't even know how people get to work out. They're like, I'm going to go work out. Like I can only do that if I accomplished everything else. Okay, so you go work out and your body temperature goes well above, right? And then you come home, take a shower, you drive 30 minutes to work, your body temperature drops, right? You go work, you have a presentation to give. What's that going to do to your metabolism? Maybe even higher than that, I don't know, but for a shorter period of time, okay? And then you're going to have lunch, your body temperature go up, sit on your butt for a little while at work, body temperature goes back down, goes up as you have a snack, goes down as you're driving home. You get home, you make dinner, you eat dinner, you watch Game of Thrones for an hour. Don't tell me what happened, I haven't watched it yet. I read all the books though, but they stopped being like the books. And then, because this is the day in which you check all the boxes, you are going to go to bed, but you're going to have sex. And then you're going to go sleep. Right? Everything got done that day. Everything. All right. So what happens over the course of the day is you are seeing that you're kind of getting the ideal range. Now, your conceptual question that you know the answer to, even if you never thought of it before. Do you ever go outside of your ideal range over the normal day? Yeah, you do. Does that indicate disease? No. So if my body temperature went outside of the normal range, when is that an indication of disease?
When it stays outside.
Yes, exactly. So everybody pops out of the normal range, right? But a healthy person with normal homeostasis is going to pull back into the normal range as soon as that is no longer serving you, right? Does that make sense? So you will always pop out of the normal range on both ends, and then you'll bring it back up. So what negative feedback is doing, do you see? Anytime it gets too high, you will, as soon as it is reasonable to do so, bring it back down. As soon as it gets too low, you'll bring it back up, right? So a healthy person will always go outside of normal range. The indication of health is not that you don't ever go outside of normal range, because that means that you never did anything, but that you come back in. Has anybody ever had a cardiac stress test besides me? Okay, so, yeah, treadmill? EKG. This one's running on a treadmill? Okay, so I have a heart problem. And I got it because I had rheumatic fever when I was a kid. And so they do cardiac stress tests every once in a while to see how my heart is doing. And so what they do in a cardiac stress test is kind of directly related to this. So they have you run on a treadmill, and as you run on a treadmill, they're testing, of course, your blood pressure and your heart rate and your O2 sap to see how much oxygen is getting to your tissues and that kind of thing. And so what they're looking at not just is like what your heart does when you're on the treadmill, but more importantly, what it does when you stop, right? So one of the indications, the good indications of a healthy heart is that, yeah, my blood pressure goes up when I'm on the treadmill, but what does it do as soon as I get off? It goes right back down, right? So that's not hypertension. Hypertension is when your blood pressure goes up and stays up, right? Especially when you're not running on a treadmill, good? So what this is going to do, negative feedback over time, tends to vacillate in and out, but very near an ideal range, right? And your range is a little different than mine, but because we're the same species, it's going to be pretty damn similar to mine, right? Okay, all right, so now the next thing is how. How do you do this in the human body? So we know that homeostasis is the goal, and we know that negative feedback is the mechanism. Yes, when it goes up, you bring it back down. And now what we need to do is put it together with some body parts that can actually help us accomplish that. And the body parts are called collectively a reflex arc. So let's conceptualize what we mean by reflex arc. So let's talk about body temperature still, because it's a really easy one to assess for homeostasis. So with body temperature, in order to maintain my body temperature when it goes up, what I have to do is I have to have the capacity to measure it, right? I have to have the capacity to send that body temperature measurement to a place in the body that knows what my body temperature should be. You guys with me? So I have to have a sensory receptor, okay? And then I have to have the ability to send it somewhere. So let's start here. In this particular instance, the stimulus is going to be a change in body temperature. Yes? Okay. Some measurable variable. You're not measuring it externally, you're actually measuring it internally. So the stimulus is a detectable change in the internal environment this time. Okay. But measuring it does not fix it. Do you agree? Okay. So I've got the stimulus and I've got the sensory receptor that says, hey, suppose I prefer 98.8 and it is 103. Right? Because I'm running outside in the middle of the day, okay? So now what happens? I haven't fixed it yet. I have to involve other body parts in order to do that. So the sensory receptor says 103, oh crap. And what it does is it sends the information via an input pathway, also called an afferent pathway. You guys remember afferent and efferent. Generally nervous system pathway, although you can use hormones if you're not in a hurry. Hormones are not that fast, right? An input pathway to the place in the body that knows what your body temperature should be. That place is called the integration center, generally. Does anybody know what the integration center for body temperature is? It is the hypothalamus. In phys, if you can't figure it out, guess hypothalamus. Honestly, because that thing, it's so anatomically unimpressive and it does like every damn thing physiologically. So it is the hypothalamus. The hypothalamus has the ideal temperature and also the acceptable error signal stored, okay? So what it says is the hypothalamus has somehow stored that I prefer 98.8, and then it gets an information that it says, okay, we're not at 98.9, we're at 103. And then it says that error signal is big enough that I actually need to institute a change, right? So here's what the hypothalamus does. Hot. How hot? Too damn hot. Needs to initiate a change. So the next thing is, you haven't fixed it yet, right? So now you have to do the output pathway or the efferent pathway. Where is the hypothalamus going to send signals? To any place in the body that can effect a change in body temperature. Yes? Those are called effectors. Okay? Now it's different for different stimuli. What are the effectors that could help with lowering your body temperature? Sweat glands for sure. Yeah. And then what about blood vessels? Couldn't you do vasodilation to the surface, right? Cutaneous vasodilation, that would help. In addition, you can also send it back to the conscious portion of your brain to say, sit down, you're hot, damn it. Also drink some water, okay, because it's too damn hot. You know it's going to get cooler. Why did you run in the middle of the day? Okay, that makes sense? Okay, so anything that can effect a change. So those are the effector organs. And then ultimately what happens is you get a change in the variable. That's the response, right? So the body temperature was too high, and now I drop the body temperature lower. But this will not help at all unless you feed that response back in as input. Because do you see, if you don't feed the response back in as input, you will continue to use those effectors and create the opposite problem. That's why this is a reflex arc. Feed it back in as input. So now I continue to feed the information in, and now I went, okay, from 103 down to 101, down to 98.9. And then can I stop sweating, potentially? Yeah, I can probably stop sweating. The rest of it will probably happen because of the moisture that's already there, and it's cooling down. I feed it back in, and it says everything's cool now. Right, does that make sense? So it's a reflex arc. So let's put this all together for you because there's three related terms. Homeostasis is the goal, yes? Negative feedback is the mechanism. Reflex arc is the parts that allow for all that. Does that make sense to you guys? Say they're all related. Homeostasis is the goal. Negative feedback is the mechanism. And the reflex arc is the parts that can accomplish it. Good? Okay, so concept check question for you. Do I need to do all parts of the reflex arc all the time? So let's say I'm monitoring my body temperature and trying to keep it in the ideal range. Do I just, for instance, check it every 30 minutes, right? Or which of those do you think that I am constantly doing of the five?
Sensory.
Constantly sending a new stimulus to a new sensory receptor. Do I stop there, or do I also have to send it via the afferent pathway to the integration center. Right? I always have to do those first three, don't I? Right? Do I always have to do the other ones?
No.
Only if the error signal is big do I have to do the other ones. Now, conceptually, why do you not just take your body temperature every 30 minutes?
A lot can happen in 30 minutes.
Yeah, exactly. A lot can happen in 30 minutes. You could have fallen into a boiling vat of oil in 30 minutes, right? I don't know that this would help you in that situation. Or if you're a parent, have you ever experienced how fast a baby's temperature can spike? Yeah, so it basically you are assessing these constantly. Does that sound expensive to you?
Oh,
To constantly assess and send information and check to make sure it's okay, right? So this is part of the reason homeostasis is so very expensive, because in order to keep the system in homeostasis, you have to be constantly checking that it's not leaving homeostasis. Does that make sense? Okay, so this is a reflex arc. We will see lots of examples of reflex arcs today. You can, of course, see this in your home with your air conditioner, right? That's a phenomenal example of a reflex arc, isn't it? So your AC has a number stored, 72, right? Door opens, goes up to 73. You're constantly assessing that information. So you're taking the input, which is the temperature, and you're using a sensory receptor, which is just your thermometer portion of your thermostat, right? And you're sending it to the brain of the thermostat. And the thermostat says, hot, how hot? Is it too damn hot, or is 73 okay? 73 is okay, right? But then it does that again. And you can see at home why you wouldn't want it to do it every 30 minutes, because you know what can happen in 30 minutes, right? Kids leave all the damn doors open in 30 minutes, right? Yeah, they were born and born, but so were you. That's what parents don't remember. I remember now my 16-year-old is a monster. And I'm like, oh, I have so many regrets. Why? Why was I a monster too? I was. I was the sassiest child. And then I'm like, where did you get it? I don't know where she got it. Okay, so now let's talk about something that sounds like it's the opposite of negative feedback, but it's not. And you may have not met this concept before. Okay, so a feed-forward mechanism sounds like it might be the opposite of negative feedback, but it's not. Feed-forward mechanism is what we call anticipatory homeostasis, because sometimes you do things so regularly that your body can predict what might be needed. So let's say you're one of those super duper regular people who gets up at 6:30 every morning and goes to bed at 11:15 every night. Day in, day out, day in, day out. You're super duper regular. And what will happen to you if you forget to set your alarm? In all likelihood, you're going to wake up at 6:20 in the morning, right? What's going to happen to you if you decide you're 21 again and you're going to pull an all-nighter? What's going to happen at 11:15? You're like, oh, dude, I'm out. Sorry, done, right? How about when you go on vacation? And you're like, I'm going to sleep all damn day. What time are you going to wake up? You're going to wake up at 6:20, okay? This is because when you are really regular, your body says, hey, every single day, around 6:20, we need our metabolism to increase because we got crap to do, right? So what they do is right before 6:20, it's like, this would be super helpful. So I'm going to increase your metabolism, which is why you wake up, right? And every night, it's like, you go to bed at 11:15, I'm going to release melatonin at 10:45 to make you go to sleep at 11:15. So anticipatory homeostasis is when you get the benefit of regularity. And if it's right most of the time, then it's going to keep doing it. I am not a regular person at all. I don't go to sleep at the same time every night. I don't wake up at the same time. My husband's a night owl. My kid's an early morning. I love both of them. So I just generally am sleep deprived. And what happens if I don't set my alarm? I am going to sleep probably all damn day if I don't set my alarm. So anticipatory homeostasis only helps when it's beneficial most of the time. Does that make sense? A couple of other examples of anticipatory homeostasis. You walk into grandma's house, you smell chocolate chip cookies. What does your anticipatory homeostasis do? It is going to start salivating, because the next thing I generally do when I have this smell is need to break down carbohydrates with salivary amylase, right? But grandma smacks your hand and she says, that's for Relief Society, right? And then you just wasted calories. So you need to tell her, I wasted calories, I need a cookie, grandma. Or sexuality, right? So let's say you have been married for less than 10 years and your wife comes out in lingerie. you might have an anticipatory homeostasis response, right?
That's what it's called.
And then she's like, oh, I was just trying it on. I got to go. And you're like, damn it, I wasted calories. But she doesn't owe you anything, not even a cookie. Okay, so let's talk about types of negative feedback. Okay, so let's talk about types of negative feedback. So we're trying to accomplish the same thing. We're trying to stay in or get back in the ideal range. But there's a couple of different ways you can do it. I've got extrinsic regulation and intrinsic regulation. Both are trying to accomplish the same goal, which is homeostasis via negative feedback. Now, I'm going to give you a couple of descriptions, and you're going to match the word. Okay, extrinsic and intrinsic. One of those two is if I have a disturbance in a body system, I handle the problem with the body system. Which one do you think is that? Intrinsic. And the other one, I have a disturbance in the body system and I am not capable of handling it myself and I have to call in the troops. And that is extrinsic. Yes. Okay. So let's look at examples of each. Okay. Extrinsic is generally longer distance, usually not quite as fast. And the troops, you usually call them in using the nervous system or the endocrine system, which are your communication systems. So if you see a body system where you create a problem, then you call in the nervous or the endocrine system, it's generally going to be extrinsic. So I'll give you guys a couple of examples of extrinsic. Okay, I'm going to create a problem with the body system and not be capable of solving it with the same body system. Okay, here's the problem. You are my 11-year-old son, and you get 6 full-size Butterfingers at Halloween. And you have a 16 and a 17-year-old sibling. How many of those Butterfingers do you eat? You eat 6 of them, because that's the safest place to keep the damn thing, isn't it? Right? And so it is going to severely disturb the homeostasis of your glucose levels. He's not diabetic, don't worry. Okay? Now, what's going to happen is I created a problem with the digestive system, but I literally cannot poop it out fast enough to solve that problem. Right? I mean, you could throw it up, but I don't want him to do that. Okay, so what's going to happen is I've created a glucose concentration problem. And I can't solve it with the digestive system, so I need to call in my friend the endocrine system to help me. Glucose level is way too high. Here's what happens. The pancreas assesses and detects the glucose level, so it's both the receptor, it is also the integrating center because it also knows what acceptable glucose levels look like. And the pancreas says, too damn high. And what it does in response is releases a hormone called insulin. And insulin is now involved in the output pathway. You guys with me? Okay. And insulin goes to effectors almost everywhere in the body. Almost every cell in your body can respond to insulin. And does anybody know what insulin does to cells regarding glucose? Causes them to suck up the glucose, right? And what will that do to your glucose levels? Drop it. Feed that lower glucose level back into the system to know when to stop releasing insulin. Does that make sense? Okay, so 2 questions for you. Do you see that this is an example of negative feedback? Can you see that? Glucose level is high, you brought it back down. And now somebody, in your words, not mine, tell me why it's an example of extrinsic regulation. I promise I'll be nice. When you're brave, I'm nice. Go ahead.
Because the pancreas is part of the endocrine system?
Yeah, pancreas is part of the endocrine system. Yeah, so it's also part of the digestive system, but not in this particular context. Go ahead.
I was just going to say it's macro. Because the digestive system can't handle
No.
all the glucose, it has to go, okay, well,
I
now
need
we're just
help.
going to address everything else.
Yes, exactly. So it's going to call on something that actually can handle the glucose level. Does that make sense? Okay, so I just want to give you the opposite example so that you don't think that this is negative feedback because the glucose level went down, because that's not why it's negative feedback. So let's talk about fasting, because this is another example. So I'm not from around here, but I've heard that people fast here because of religious reasons. Sometimes I have to fast because my doctor says that I need fasting blood glucose levels, right, and cholesterol levels. And then I go in and I have an appointment at 10, they make me wait until 10:45, and you're going to eat the damn doctor at that point, right? You're like, I'm literally going to eat you. So that is a disturbance of homeostasis that you voluntarily or not created via not taking anything into the digestive system. And you can't or you won't solve it with the digestive system, whatever, right? So now I'm going to have fasting, which is the disturbance of glucose, because my glucose level will be really low, right? Okay, so I'm not going to eat. How am I going to solve this problem? In this particular instance, again, your glucose level is detected by the pancreas. This time, the pancreas says the error signal is too high, too low the other way, right? But it's definitely not going to release insulin, because would that help? No, it'll make it worse. It is going to release a different hormone called glucagon, which is easy to remember because you need it when your glucose is gone, okay? And that's going to be the output pathway, but now it's going to go to different effectors. It's going to go to the places in the body that can help because they stored glucose in times of plenty. Anybody know what those might be? Liver is the primary one. So the liver has glycogen stored, right? And glucagon goes to the liver. And what's it going to tell the liver to do? Dump that glucose into the bloodstream. And then what happens to the glucose level? It goes back up. So do you see that this is an example of negative feedback, even though the variable is going up? Yes? And do you see that this is also extrinsic? Digestive system problem that you created, right? And handled with the endocrine system. Does that make sense? Okay, so sometimes you can feel this, by the way. I am a person who tends to not eat frequently enough. I'm just like, I forget, and then I'm busy, and it's a pain in the butt.
And then
So,
you eat the house.
what?
And then you eat the house.
Yeah, no, then I have no appetite whatsoever. But I get really stupid and kind of mean when I don't eat. Like I can't figure out how to work a doorknob, and I want to yell at it to get the solution. I'm like, ah! So, like, what is happening? Why am I in the kitchen? Sometimes I'll cry, and my husband's like, here's a cookie. See you in 20 minutes. But sometimes you feel this, right? You like have been hungry, and you're too busy or whatever, and you're getting kind of dumb because your brain primarily runs on glucose, and sometimes you're getting kind of pissy if you're me. And then all of a sudden, you still haven't had time to eat, but all of a sudden you're fine. You're like, oh, I'm fine. What is that? Glucagon. That's what that is. Okay? You're going to run out of glycogen pretty quickly. So when that happens, you still need to go feed yourself. That also, you can also go into ketosis. It'll make your breath smell weird when you haven't eaten often enough. You know that, right? When people are dieting, it makes your breath smell weird. Okay, so those are two examples of extrinsic. Now let's do two examples of intrinsic. Intrinsic regulation is when I have a problem and I handle it in-house. The metaphor that I will use for intrinsic versus extrinsic is somebody robs my house, I call the cops. Extrinsic regulation. Can't really manage it in-house. Somebody robs a gangsta's house, either a gangster or a gangsta, right? Do they call the cops?
No.
Hell no. They're going to handle that crap in-house, right? Whether it's Tony Soprano or somebody in Compton. Doesn't make any difference, right? They're going to handle it in-house. That is intrinsic regulation. Does that make sense? Okay, so let's go look at a couple of examples of intrinsic regulation. So sometimes, by the way, intrinsic is also called auto-regulation or local regulation. Auto as in self, local, obvious, right? Okay, what do you predict about whether intrinsic will be faster or slower than extrinsic? Faster. How about robust? Do you think it's going to be able to make as massive a changes as extrinsic? No. It's really going to be able to make small local changes, but usually relatively quickly. Okay, so let's look at an example. Let's say cells in one tissue but not the whole body need more oxygen. If the whole body needed more oxygen, we could increase respiration rate and increase heart rate, right? But what if it's just one tissue? Here's an example of why that would occur. You went on a bender last night and you had 6 shots of tequila. What is your body going to be doing the next day? Laying on the couch, regretting your poor decision-making skills. But what is your liver going to be doing? Working its little liver butt off, right? It's like, I got some work to do, people. The same thing will happen if you were ill yesterday and you took too many acetaminophen or Tylenol, right? It's just as hard on your liver. So by the way, never take Tylenol when you've been drinking because they're double whammy on your liver. Always take ibuprofen or aspirin, okay? Either way, whether you did it the fun way or not, you are going to be laying on the couch and the rest of you does not need much oxygen, comparatively speaking, and your liver needs boatloads of oxygen because its metabolic rate is going crazy. So here's what happens. The liver needs oxygen, and what it does is the liver cells, as soon as their oxygen level starts to fall, will release chemicals locally that will cause local arterial dilation. How does that solve the problem? Increases blood flow and oxygen flow into the area, correct? Is it going to massively change the chemistry or pressure of the rest of the body? Might have a teeny tiny impact on blood pressure, but nothing really demonstrable, really. Okay, so, and what's going to happen is as soon as you've got adequate levels of oxygen, it'll stop releasing those chemicals, right? So, do you see that this is an example of negative feedback? Yeah? Do you see that it's intrinsic instead of extrinsic? Does that make sense? Okay, so, now the next example is a little harder to grasp, but I'm going to teach it to you because you're going to see it all semester long. And just, take the pill early. Just get it over with, right? Nobody likes it. It's fine. It's going to torture you all semester long. So every once in a while, you're going to have to memorize a chemical equation. And this is going to be one of the few that you have to memorize in phys. Okay, so speaking of metabolism, we know that as the metabolism of tissue increases, not only do you use more oxygen, but you generate more carbon dioxide, correct? Okay, now, carbon dioxide has a potential impact on pH, which is one of the reasons we have to keep our carbon dioxide levels regulated. And here is the relationship, okay? So cells or tissues are generating carbon dioxide because of their cellular respiration. But CO2 will combine with the ubiquitous H2O. And of course, as you get more active, more carbon dioxide. What is the problem here? The problem here is with carbon dioxide and water combined, you generally make carbonic acid, and carbonic acid does what acids do, which is it dissociates to an H plus, okay, and HCO3 minus. Okay, which one of these players will change your pH? H plus. Okay, so let me tell you the relationship. Here's the relationship. As you increase carbon dioxide, you tend to increase H plus. And what does it do to your pH? Lowers your pH and makes it more what? Acidic or alkaline? Acidic. Okay. Now let's get to how this is an intrinsic regulation issue. So as soon as your gas levels start to change and your pH levels start to change, what happens is, without even telling the nervous system or the endocrine system, as that blood with a lower pH and a higher carbon dioxide level starts to go through the lungs, it will immediately trigger an increase in respiration. Do you see? And what will that increase in respiration do? It will blow off carbon dioxide, right? Which will drop your H plus level, which will raise your pH. Does that make sense? Okay, so you didn't have to call on another body system. You naturally have these chemoreceptors in your cardiovascular and your respiratory system, and this one's respiratory. And as soon as you detect an increase in carbon dioxide or a decrease in pH, you will increase your respiration rate. Is that good? Okay, all right. So you are going to see this equation all semester long. By the way, for those of you who have trouble with pH, and a lot of people do, because some knucklehead decided that pH was the negative inverse log of the concentration of hydrogen ions in a solution, which makes you have to do math before you can even think through the problem. You just need to have a good enough understanding of pH to know if you increase H+, what's going to do to pH, and is that more acid or alkaline. If you are really uncomfortable with that, of course it's in your textbook, but also in interactive physiology, they have a good pH review for you. So most organs have the capacity to do intrinsic and extrinsic. And what they will do is the body has a habit of doing the cheapest, fastest thing first just to see if it will work, right? So if in that liver situation, right, you're laying on the couch, you try intrinsic regulation and you release chemicals that cause arterial dilation, and that still didn't get you enough oxygen, then you would try extrinsic. And you would tell the body, right, the oxygen level is too low, and you could increase your respiration rate and increase your cardiovascular system rate. Have you ever been so hungover that your heart rate was high and you were breathing heavily? Probably in Vegas, maybe? I don't know. Okay, you guys are all right with that? Okay, so positive feedback. Positive feedback is the opposite of negative feedback. It will not maintain homeostasis. So positive feedback is not homeostatic. It's also not anywhere near as common as negative feedback. So with positive feedback, I want you to dump your connotations of the word positive, because it's not necessarily good. As a matter of fact, sometimes it's bad. And it's not necessarily up. But what it does is where negative feedback, wherever the variable is going, negative feedback will negate it. Wherever the variable is going, you're going to add to it. So for instance, if it started getting hot in your house and you turned on the heater, that's a positive feedback situation, okay? So what can happen is if you were in a situation like this and you were getting into a positive feedback loop, if, for instance, I had a fever and instead of sweating to cool down my body temperature, if I was in a positive feedback loop at that particular moment and I started shivering, and that's probably happened to all of you, right? And what happens is body temperature was going up and then you added to it, correct? Okay, so what's going to happen at the end of that? Well, if you're otherwise healthy, you know that you don't die from every damn fever you get, right? So you go into a positive feedback amplification, and then what happens is you take an Aleve, you take a Tylenol, you take an ibuprofen, or you just get over it, right? And eventually you get a hold of it and pull it back into negative feedback, right? But as long as you're adding to the problem, you're in positive feedback. Does that make sense? Okay, so a lot of times positive feedback situations are disease situations. Now, if you are otherwise healthy, you don't generally die from those situations, right? But sometimes you can need an intervention. If you are at the extremes of age, very tiny people and very old people, if they get into those situations, they are less likely to catch it without intervention, right? So teeny tiny little people, right? Their fevers can spike really quickly, and they can go from 99 to 104 or 105. And are they going to catch it? No, you're going to need to intervene, right? Or else they're going to go into seizures, febrile seizures. So you do what parents do or what medical professionals do. You intervene with an analgesic that can also a fever reducer, right? If that's not going to be fast enough, what do the parents have to do? You dump them in a cold bathtub, and then everybody hates everybody, but they live, right? They live through the situation. So the very elderly have that problem too. If they get very cold, it's very, very hard for them to warm up. If they get very hot, it's very, very hard for them to cool down. And you almost always need to intervene. Another example of a positive feedback situation is if I nick an artery, what's going to happen to my blood pressure? It's going to drop. And my heart thinks it's helping. But as my blood pressure drops, my heart's kind of a one-trick pony, and it's like, hey, I know what to do about low blood pressure. Let's increase the heart rate, which is going to make me lose more blood, right? Which is going to make my blood pressure drop, right? It's just like the heart is like not super smart. It's like your beefy, meaty friend who like has one trick. Can I punch it? So it tries to punch and it doesn't help, and that's a positive feedback situation as well. Now, with positive feedback situations, what tends to happen is you go along with negative for a while and then you lose track of it either up or down. That's what happens with a positive feedback situation. And they're also often called cascades or amplifications or positive feedback loops. Now, there are a couple of healthy ones in which it's not a disease situation. And when you do positive feedback and it's not a disease situation, it's usually because once you started that, you really want to finish it quickly. Birth is a great example. Okay, so labor. Nobody ever said, I sure wish I was in labor for longer. Okay, so what happens is when the baby is full term, assuming that the baby gets to decide when the baby's going to come out and not the doctor who has a vacation planned, right? When the baby is full term, they will release oxytocin, which will cause uterine contraction. And the uterine contraction will cause the release of more oxytocin, which will cause more and frequent uterine contraction. And they continue to get stronger and closer together and stronger and closer together until boop, right? Because you didn't want to be doing that all damn day, right? So that's a positive feedback situation in which it's healthy, right? It ends in an event. Blood clotting is another positive feedback situation. Let's say this time I nicked a small vein instead of an artery. What happens is that attracts platelets to the area, which start clotting, which attract more platelets to the area, which start more clotting, right? Until you plug the hole. Because you don't want to fart around with that either, right? Okay. And then the last example, you probably are not familiar with it yet to know that this is a positive feedback situation, but in like 2 sets of notes, we're going to be doing action potentials. And action potentials is how you get an electrical stimulus to go along a neuron really, really quickly. So what you do is once you start it, you want it to go as quickly as possible. So we use a positive feedback situation in that too. That, okay? So even if you're not familiar with that, I'm going to put it there so that I can refer to it later and remind you that you learned that it was positive feedback. Estrogen is also a positive feedback situation. Okay, but most positive feedback examples are disease situations. They are vicious cycles, and they potentially lead to death. And a lot of times you need intervention to stop a positive feedback situation. Like, what could stop me from dying in that nicked an artery situation? Yeah, put pressure on it. Plug that damn hole. Do something, right? Tourniquet, pressure, whatever the hell you need to do. Okay, that makes sense? Okay, so just a little bit more, and then I'll let you guys go because the other class is slightly behind you. So coming back to the theme of homeostasis, which is what we've been trying to accomplish the whole time. Homeostasis is the maintenance of a relatively constant internal environment. And it is the predictor that you are going to use for phys. So like if you want to figure out what's going to happen, what would maintain homeostasis? If you disturb it this way, I predict that it's going to do this. And so what we're going to see is with the cardiovascular system, you say, okay, I have a situation in which my blood pressure goes up. What I predict is that my blood pressure is going to go down. How? By any means necessary. So what is consistent with bringing your blood pressure down? That's going to be part of the phys that you understand. For instance, would decreasing your heart rate help to bring your blood pressure back down? How about vasodilation? Yeah? How about peeing out some fluid? Right? So what would you do? Yes. Okay? Generally speaking. And some of them are faster, and some of them are slower, and some of them are long-term solutions, and some of them are short-term solutions. But that's basically what you use as your predictor. So homeostasis is the unifying theme of physiology, and it is primarily, as we said, the ECF that you monitor and regulate. And for instance, we are regulating every damn thing that you can imagine. You are right now working your butt off, right? You should get the day off because right now you are regulating your body temperature. You're regulating your pressure. You're regulating your pH. You're regulating your glucose concentration, your sodium, your potassium concentration, your body temperature. All of that is what you are doing right now, baseline, right? And that's why it requires about 60% of the ATP. The 60% of the ATP is just keeping you from going toward entropy, right? And entropy in humans is death, right? And entropy in animals is death, right? Okay, so what we're going to do next time is we're going to talk about what does constant mean. When I say maintaining a relatively constant internal environment, you know I don't mean 98.6, right? So what do I actually mean? What I mean by constant is what sounds like an oxymoron. What I mean is predictable variability. Sounds like I don't know what the hell I'm talking about, but I do, right? It's variable, but in a predictable way, right? You know that your body temperature is going to vary, but you know how it's not going to vary, right? Over the course of a healthy day. Okay, so I will see you guys on Thursday. Your quiz won't be until Tuesday.
I'm sure this is in the syllabus, or you already went over it, but how many questions are in a given quiz?